If it's hump day, it's hemp day. Welcome to the Hemplet Farms webinar series. I'm Robert Colangelo. I'm founding farmer of Hemplet Farms, and we're a high-tech propagation company. We're located in Portage, Indiana, and we provide high-quality seedlings and propagation services. And I'm going to be your moderator for today's webinar. The goal of our webinars is to create a forum to exchange reliable, trustworthy scientific information and share experiences with like-minded professionals in the Midwest hemp community. Whether you're a grower, processor, handler, or retail seller, or you're in government or academia, we all share a common goal to promote hemp so that we can all prosper in this new emerging market. I want to give a big thanks to Christian Lee and the Hemplet Farm teams for helping us put this together. Marguerite Bolt at Purdue University Extension Services, Bree Sierra at Newton County Soil Conservation, and the Midwest Hemp Council. I'm really excited today. I had the pleasure of visiting our speakers' Davis, California offices last month, and I was very impressed with what they're doing with genetics and breeding, and I thought it would make for a great presentation for our webinar series. So I'm going to start out with what is speed breeding and how can it benefit the hemp industry. Much of the current hemp genetics are unstable, as a lot of us know. They exhibit a wide range of genetic variation and traditional breeding techniques to get the seeds we desire can take as long as 8 years to find a breed that's stable with desired traits. Speed breeding is a faster way to bring stable seeds with select traits to market, and they use efficient and versatile gene editing technology like CRISPR-Cas9 to modify, delete, or correct precise regions of hemp DNA and be able to produce seeds sometimes in less than 3 years. Our original speaker had a conflict and wasn't able to make it today, but we've got even a better speaker, and that's Jeff Mills, the plant breeding lead at Green Venus. His role is to lead breeding efforts in hemp, raspberry, lettuce, and tomato sectors. Prior to joining Green Venus, Jeff spent 19 years in R&D at Seminus Vegetable Seeds. In those years, he leveraged many breeding tools to bring innovative new melon varieties to the market. Jeff's passionate about plants. He really believes in this, and he does it at home when he's done breeding at work. And he is excited about marrying traditional breeding techniques with modern gene editing technology to accelerate the development of crop plants that benefit growers and consumers. So with that, I welcome Jeff Mills. Jeff, it's all yours.
Thanks, Robert. Thank you for the opportunity to do this. I'm really looking forward to it. So today I'm going to give you a brief introduction to who Green Venus is. I'm going to talk a little bit about our 3 technology platforms and how they apply to developing new varieties. I'm going to make a case for why anyone should bother to breed hemp. I'll talk about some breeding methods, traditional breeding methods that have been used in cannabis over the years. And then I'll give you a brief introduction to gene editing techniques and how they might apply in agriculture, and then compare and contrast traditional breeding with gene editing, and hopefully solicit some ideas from the audience as to breeding targets for the future. So who is Green Venus? We have approximately 50 very dynamic employees. Our R&D hub is in Davis, California. We also have employees in Florida and Arizona. We have an R&D team with expertise in molecular biology and genomics, bioinformatics, plant tissue culture, plant pathology, plant breeding, and agronomy. And you can see a picture of our team, but unfortunately, due to COVID, we don't have a recent group picture because we can't crowd everybody together in one place. I'm hoping the team's changed and grown quite a bit over the last year, so I'm hoping to get a new picture soon. We have 3 kind of discrete technology platforms, and I'll briefly describe each one, and then we're going to go on and focus more on the first 2. So micropropagation is what we refer to as our Botticelli platform. So this is a proprietary non-GMO propagation technology. It's essentially plant tissue culture, right? Where we can propagate large number of plants from a small amount of starting material. And we've focused this platform on cannabis, avocados, and berries, but certainly it's applicable to lots of other species. So gene editing, we refer to as our Primavera platform. So this is gene editing using Cas9 or similar enzymes. And this platform enables speed to market with non-GMO traits that benefit growers and consumers. We are applying this platform in lettuce, tomato, avocado, hemp, and berries. And I'll talk mostly about hemp today. I will talk briefly about lettuce because it's our first commercial product. We just launched it last year, and it's a good example of an application of the technology. The third platform is Florian. So this is novel technology for hybridization. Depending on how it's applied, it may result in a product that's regulated as a GMO, or it may be non-GMO, depending on the technique used. And it's largely focused on row crops. We do have work on hybrid lettuce as well, and that would be non-GMO. But I'm not going to talk a lot about Florian today. We're going to focus on hemp, which means micropropagation and gene editing. So challenges in the hemp industry. And when I say challenges, really, I'm going to list challenges, but I'm also listing the laundry list of things that a breeder needs to be thinking about and trying to address for the grower. So this is my case for hemp variety improvement. Seed quality and consistent genetics are definitely still an issue in hemp. So growers struggle with non-compliant THC levels, the wrong chemotype showing up in the field. So finding type 1 or type 2 plants in hemp seed, males showing up in seed lots that are supposed to be feminized, and a lack of uniformity in the varieties. So you stand and look at a hemp field and every plant looks different cannabinoid ratios, different sizes, architecture, which can make just planning for the crop and planting density and weed management, harvest, all these things become more challenging if you don't have a predictable plant performance in the field. A need for regionally adapted varieties. The industry is so new that there really are very little is known about regional adaptation of different varieties, and there hasn't been enough time to develop varieties that really excel in specific performance, excel in their performance in specific environments. So there are pretty large differences between hemp growing regions in latitude and day length, and different varieties behave very differently in different locations. Different needs for disease resistance and adaptation to temperature and humidity that can vary widely across the country. Need for varieties that create value for the grower. So really, we need to focus on traits that make the grower more profitable. So novel cannabinoids that bring a higher price to the grower, improved grain or fiber quality, improved harvest index, labor reduction traits. These are the sorts of things that, moving forward, hemp breeders need to be thinking about. Okay, so why hemp? Green Venus works on quite a few different crop species. We chose hemp because it's a relatively young industry. We see a lot of opportunity for variety improvement and a lot of opportunity for growth in the industry. There's so many different uses for hemp, many of which are really still underutilized. Seeds for food and oil, stalks for building material and textiles, and of course, cannabinoids from flower and leaves. We just see it as kind of an untapped resource and possibly a very profitable crop for growers to move into as the market develops. So traditional breeding in cannabis. I'm going to talk through 3 different traditional breeding methods that have all been used historically in cannabis, just to orient you to what traditional breeding looks like and how to see the roadmap that led us to where we are today in hemp genetics. So breeding strategies. So this breeding strategy is essentially population development followed by clonal propagation. And this has been the standard breeding method for high THC cannabis up until just recently. even still today. So this technique, this method would be really similar to what's done in other clonally propagated crops. So berries, fruit trees, strawberries, raspberries, things like this. Very similar breeding method, right? But it's easy to imagine how this evolved, right? You have these non-structured populations. You don't do a lot of self-pollination. You essentially grow out a lot of seeds. You select the handful of individuals that have the traits you're interested in. You clonally propagate them, and that's what you grow. So you then take your favorites of those and cross them to your other favorites that may share traits, or maybe you think they have some complementary traits. But because you're always out crossing, you're never self-pollinating or sibmating, right? Crossing two related individuals. You're typically crossing unrelated individuals from different populations, and you're not doing a lot of self-pollination. So you're rarely fixing traits. So these seed lots are always really heterogeneous, right? There's a ton of variation in every bag of seed. And this is kind of where the high THC germplasm, it's how it's evolved, and it's still how it's being bred. And it works well for a clonally propagated crop. But if you're a hemp farmer and you're planting a seed crop in the field, having wild lack of uniformity and individuals that are all over the map in your field is really a challenge. It's a problem. A lot of the modern high CBD, high cannabinoid hemp germplasm is derived from marijuana germplasm. There are a couple of alleles that need to be switched over in crosses to hemp, but largely the background leans quite heavily towards high THC cannabis, and it's for good reason, right? We're growing for cannabinoids, and those are the backgrounds that have been selected for high cannabinoid production. So we've kind of inherited these breeding methods that lead to a lack of uniformity in the field. Okay, so this is the next breeding method. So this is mass selection. And this or something like it is largely how land races were probably originally domesticated. So as growers first began to grow hemp out of, selecting out of wild populations, early domestication probably happened through a process like this, where essentially you plant a seed lot in the field, males and females all mature, open pollination takes place, right? So every female, the little pink arrows in here, was pollinated by multiple males in the field. At the end of the season, you'd select the best performing females and you'd combine the seed from those, and that would be what you'd plant in the following year. So there is some selection happening, and there is a moderate reduction in heterozygosity. So over the course of many years of repeating the same process, you are developing a more uniform variety over time, but relatively slowly. Traits that are controlled by one or two genes, you may end up fixing and not seeing variation for those traits anymore. Traits with more complex inheritance that involve multiple genes probably are still segregating the population, but hopefully you're nudging it in the direction you want. This technique is or something like it is probably still used pretty heavily in fiber and grain hemp breeding. Those varieties tend to be open pollinated, and some combination of self-pollination and mass selection like this is probably how we got to many of those varieties. So hybrid pedigree breeding. This is typical in maize, in field corn, and in most vegetable varieties, hybrid vegetables. Typically, this is how new varieties are developed. So essentially, you have multiple families in your breeding program, and they may be at various levels of inbreeding. But you make a developmental cross between two individuals, right, to generate the F1 generation. That plant is self-pollinated or sibmated, and you harvest the F2, where you see wide variation. And the breeder selects individual plants and either self-pollinates or sibmates them to a related individual multiple times over many years, right? And so after many subsequent rounds of self-pollination, you've really inbred that line to the point where it's quite phenotypically uniform, and you know what to expect from it. When you get material that is then fully inbred, you make crosses between those inbred lines to produce F1 hybrids. And those F1 hybrids are what would be sold commercially, right? So these are benefiting from heterosis because you're crossing unrelated inbreds. So you see yield and performance advantages in an F1 hybrid, and they're quite uniform as long as the parents were sufficiently inbred. The caveat to this in hemp is that you have to balance inbreeding to improve uniformity against inbreeding depression. So in some families, you'll see some degree of reduction in vigor and performance as you inbreed. And so there's going to be a balance. And in different crops, this looks very different. Some crops tolerate inbreeding quite well, and others don't. But in high cannabinoid hemp, this breeding method is probably what's differentiating the better breeders on the market from the rest. And you'll see improvements in uniformity from companies that are using this strategy. Okay, so this is a fun slide, but there's a lot going on. I'll try to orient you to it. So here's me making a case for why we need breeding for uniformity in hemp. So I purchased a number of commercially available seed lots last year, and I grew them all out in our greenhouse. And we did chemotyping analysis. So we submitted samples for cannabinoid analysis to a lab. And here I've plotted total THC % on the x-axis and total CBD on the y-axis. So you'll see these two blue lines, right? And these are to orient you to what the bottom line is a 20-to-1 ratio of CBD to THC. And the top line is a 30-to-1. So you can see all these different colored dots. Each color is a variety. So all the blue dots in the upper right-hand corner, they're all from the same bag of seed. They're all sisters. All the yellow dots are from the same variety. All the green dots are from the same variety. So we have multiple varieties represented here and multiple individuals from each variety. So what I see here is clear differences between varieties, and a lot of that is probably driven by maturity, but an enormous variation within each variety, right? So you can see these individuals that are below the 20-to-1 line. They are really unlikely, those individual plants are really unlikely to be compliant at harvest testing, right? But there are other individuals from that same population that came out of that same bag of seed that were really quite competitive, and some even exceptional for their ratios. And this is a challenge for growers, right? Managing, you're essentially playing chicken as you approach that pre-harvest testing, and you want to maximize CBD production or cannabinoid production, and still you need to come in under 0.3 for THC. And so having a variety that's uniform with predictable performance provides enormous value and peace of mind. I have to go through this process for my breeding nursery as well, and it can keep me up at night, especially with a lot of different varieties all in the same field of greenhouse, and different maturity times, different ratios. It can be a challenge knowing when to pull the trigger on testing. So the next slide is actually zooming in on this population that you see in the upper right, the blue dots. So these are all individuals from that single population. So now we're looking at one variety. And this is a commercially available variety that we purchased. And the total cannabinoids here are reflected in blue. And the orange bar is the total THC, right? And the really my take home from looking at this, putting myself in a grower's shoes, there's a 2x difference in total cannabinoids between some of the low individuals that came in just north of 4 and some of the others that are at 9.5. So there's an enormous opportunity to improve, grower economics, right? Just by fixing cannabinoid production It's a huge difference between individuals, and every single one of them was different. The other thing I take home from this is that just as you saw on the other slide, the ratios are really quite different as well. We see individuals with very low levels of THC produced, right? This is probably the sweet spot, right? We have an individual with very little THC produced and close to 8% CBD, right? Given a couple more weeks, this would have probably been, if my whole population could have been this plant, I would have been pretty happy. You really hope you don't have this plant because your return is going to be quite low, and you really hope you don't have this plant. If that's the plant that you end up selecting for your, goes into your bulk for pre-harvest testing, you're not going to pass. So this is a pretty strong case for the need for breeding in hemp.
Jeff, great job at taking a complex subject and making it easy to understand. So kudos to that. Just a couple of simple questions on the mass selection and hybrid pedigree breeding. Talk a little bit about timing. How long does it take for both of those methods to get you stable genetics?
Okay. So mass selection, it's really going to depend on your population size and selection intensity. So how many individuals were in the open pollinated population and how few did you select, right? So that's, there's no single answer, but it's a relatively slow path to achieving uniformity, especially in a crop like hemp that is so prone to open pollination.
Is that
right? Because you have,
is it given in context? Is it 3 years,
let's
5
say,
years?
Let's say 10, 7 to 10 years, and the outcome is still a population, right? You don't expect perfect uniformity, but hopefully it's relatively uniform for at least the handful of traits that you really focused on.
Okay.
Yep.
And the other one, the hybrid?
The other one, it depends a lot on how aggressive you are with turning these generations. So, let's say my goal is to get to F6. So that's, I selfed an F2 plant. So that's 1, 2, 3, 4, let's say 5 generations to produce my F1. Okay. So one grower might turn a generation a year if they're doing this outdoors. Someone else might do 2.5 or 3 generations a year if they're in a controlled environment. So they're different time scales, but let's say 3 or 4 years to produce the F1. Let's say 3 years if you're being aggressive. Certainly 5 or 6 if you're doing this in the open field without the benefit of multiple cycles per year.
Okay, great. That just helps give it in context and the importance of speed breeding. So if you could go back to your scatter diagram, that one with the 2 lines.
This one, yep.
Yep. What is the average variation in total cannabinoids that you see within a variety? Is it 1 time, 2 time, 3 time?
I don't know about average. The variation in most material that we saw, it was really high. I would say we see the lowest material coming in 15 to 1 typically. I mean, occasionally you see a rare individual that's much worse, but let's say 15 to 1 with most individuals being between 20 and 30. Okay. I drew those lines really because I feel like biologically that's kind of that band in between 20 and 30 is where most material falls. But certainly you'll find individuals that are outside of that.
And last question is, what do you know about seedless triploid genetics?
What do I know about?
And how does that relate to hemp?
Well, seedless triploid genetics. I have not personally seen triploid hybrids in the open field. I'm familiar with a company that's commercializing seedless triploid hybrids, and it works very similarly to seedless watermelon, right? Where you have a tetraploid parent and a diploid parent, and you cross them to produce triploid hybrid seed that's not fertile. It's, I think, a promising area. I'm kind of interested in looking further into it, but I can't personally speak to it. I haven't seen these hybrids in the field, but I have heard about them.
So is the advantage that you would produce crop that won't go male? Is that the thought behind
Not
it?
necessarily. I don't know that you would see fewer males in the seed lot, but the advantage would potentially be that if there was stray pollen, that pollen would not result in seed formation in your crop.
Okay. To the group, I forgot to mention this earlier, we'd like to know what topics or speakers you'd like to see in future webinars. So throughout the rest of the presentation, feel free to just drop me a note on the chat function of what kinds of presentations or speakers you'd like to see in future webinars. With that, Jeff, why don't you start your second half?
Great. So gene editing. I'm going to start with gene editing 101. Kind of a quick introduction to the technology, very high level. We're going to talk about some of the practical strategies for using gene editing and its limitations. And then we're going to talk a little bit about comparing the strengths and weaknesses of gene editing with traditional breeding and kind of hopefully agree on a path forward. So Robert asked me to include this slide. As a scientist, it's not one of the things I think about all the time, but I'm really glad he did. This is often, when you're talking to a non-scientific audience, the first question, right? Is gene editing GMO? You know, should we regulate the process or the product? And that's I think an important question. So you can see I have a map here. The countries with a green check mark do not regulate gene editing as GMO unless foreign DNA from a different species was introduced, right? So these countries regulate the product, not the process through which it was developed. So if you were to develop something using gene editing technology that could have been developed through traditional breeding, right? Nothing was added, then it's not regulated as a GMO. Although you do jump through some regulatory hoops to confirm that that's the case. Countries with the red circle regulate gene editing not based on the outcome, but based on the process. So they currently treat it as GMO, even if the outcome doesn't contain foreign DNA and could have been arrived at through traditional breeding. I think the EU, I don't expect that that's settled. Currently, they're regulated as GMO. I know that many member states aren't happy about that, and I suspect it will evolve over time. Many countries in the world don't have a formal system in place to regulate gene editing yet, and potentially moving forward, it would be done on a case-by-case basis, but I expect this map to evolve in coming years. So gene editing with Cas9. So this is gene editing 101. In one slide, kind of what are we doing and what do we expect to be the result? So on the right, I have a little graphic of the gene editing machinery. I call it machinery, but I mean, really, we're talking about RNA and a protein. So we have the Cas9 protein, and really, it's just an enzyme that cuts DNA. It acts as a pair of scissors. So then we also have the guide RNA. So this is a little piece of RNA, and you can see that loop there is conserved. That stays the same, and it hooks up with Cas9. And then it has that tail that it says matching gene sequence down there. And that tail dictates where the enzyme is directed in the genome. So that little tail is going to find the genomic DNA with a matching sequence, and it's going to set the enzyme there, and the enzyme's going to make a cut. So you have breaks or cuts happening in your DNA constantly. This is happening in individual cells, in plants and animals all the time. And we have endogenous systems that repair these cuts as they happen quite quickly. And this is built into the function of our cells. Really what we're doing with Cas9 is we're making a cut, in a specific gene, right? And we guide it, the enzyme, to that gene with this matching gene sequence. It makes a cut. Normally, the cell's natural repair machinery will repair that cut correctly. But at some small frequency, it'll make a mistake when it's repairing the cut. And really, the art here is identifying those events where the mistake was made and selecting the mistake we were looking for. So this is happening naturally all the time. Plant and animal populations, there are mutations happening at some low frequency constantly. And this is where the variation that plant breeders select on occurs naturally, right? All the variation that I'm looking at when I look at a population in the field is the result of mutations, but many of them happen over a long period of evolutionary time, right? They're not necessarily recent. But gene editing allows us to increase the chances of finding a mutation in a particular gene that we're interested in relatively quickly. So in practice, this is kind of our workflow, right? So we have on the left, the blue boxes are activities that involve tissue culture, plant tissue culture. So these happen, in individual plant cells or plantlets that are grown on Petri plates, on media and sterile conditions. So we edit the cells. We need to get the editing protein into those cells, and we allow it to do its job. We then need to regenerate plants from single cells. Okay, and this is often the bottleneck when we're looking to do gene editing in a new crop that we don't have experience with, because there's a great deal of art and science involved in figuring out how to convince these individual cells growing in clumps on a Petri plate to differentiate and become individual plants. So different combinations of media and hormones, lighting and temperature, right? All the environmental stimulus, everything's important in convincing these cells to become plants. And this is one of the challenging aspects of applying this technology in a new crop. So once we have you can see our little hemp plant beginning to grow from a clump of undifferentiated cells on a plate here. Once we have that plant, we grow it up and we bring it out of culture, bring it into the greenhouse, and we would self-pollinate it. And the reason it's important to self-pollinate it, so most diploid species have 2 copies of each chromosome, which means at any given gene, you have 2 copies of that gene, 1 on each chromosome. So when we find plants that have our edit of interest, it's very rare that we would have the same edit in both copies of that gene. Typically, we only find plants that are heterozygous or have one edited copy and one what we call wild type copy, the original ancestral copy. So we need to self-pollinate these edited plants and then sift through their offspring to identify individuals that are fixed for that edit. And we can do that through sequencing. We sequence the gene and we find the individuals that have both copies edited with our desired edit. We can then do a seed increase on those individuals, and then we can start phenotyping. So we go to the greenhouse or field and we want to validate that the edit has the phenotype or the trait that we were interested in. So this is shifting gears just slightly, but this is our first commercial example at Green Venus. It's a recent product that we developed. We just launched it last year. So this is a lettuce variety that we're now selling, and it's our first gene edited product. And we were able to go from a product concept to seeing our lettuce in commercial trials in 18 months. And this is why we call it speed breeding. I'm pretty quick, but I have never developed a commercial variety in 18 months through traditional breeding. So the lettuce genome is large and complex, and there are 18 copies. We call it PPO. It's an enzyme, and it's polyphenol oxidase, and it's involved in a browning or pinking reaction, right? When you buy lettuce and there's mechanical damage or the rib is broken, you'll see you get kind of brown or pink gummy substance building up there, and it results in a reduction in quality. So we know that this enzyme is expressed in damaged cells and results in browning or pinking and reduced quality. So we went in and we identified 6 of the 18 genes that produce polyphenol oxidase, and we knocked them out. We then recovered individuals that were fixed for all 6 edits, and we began running field trials. So we've now developed a lettuce variety with better quality over time in post-harvest storage. It maintains its quality longer. And we did this through gene editing. And we were able to bring it into grower trials within 18 months. So this is just an example of one of the sorts of traits and approaches that we could take. And I'd like to be telling a similar story in hemp next year. So strengths and limitations of gene editing in agriculture. So gene editing, like any other tool or approach, there are things that it's really well suited for and things that it's less well suited for. So just to provide some context around how we might think about using this, it's relatively easy to gene edit to produce traits that are induced by a loss of function of 1 or 2 genes. So if you know the genes involved and the trait that you're interested in can be produced by turning those genes down or off, right? So they're less or not expressed. That's a great application for gene editing. Things that are more difficult to do, traits that are controlled by many genes are more challenging, right? Because you'd have to edit multiple genes and get all of those edits stacked in a single variety, which can be more complicated. Although I gave the lettuce example, we were able to do 6. So it's not a trivial thing, but it is possible. Traits that are induced by a specific change, not a loss of function, not a reduction in function, but maybe a different function or a change in sequence that's very specific. That is possible to do through gene editing, but it is more challenging. So when thinking about where to apply gene editing, you have to know the genes that underlie the trait before taking a gene editing approach. And it's very easy to break things, just like in life, right? It's easy to break something. It's much more difficult to build something. So a loss of function mutation, if that's desirable, is an easier fit than building function. Okay, so the summary of this slide is gene editing going to put me out of a job? So I'm trained as a traditional plant breeder, and that's my passion. And I joined a gene editing company. I was excited about applying a new tool to a very old craft. So I'm going to compare and contrast traditional plant breeding techniques and gene editing, and hopefully paint a picture of what makes sense for how to move forward with variety development. So traditional plant breeding is great for complex and quantitative traits, so traits that are controlled by a large number of genes, and especially a large number of genes, maybe each having a small contribution. Traditional plant breeding is a better fit for those traits. Genomic information can be really helpful with traditional plant breeding, but it is absolutely not required, right? I don't have to have any sequence data to stand in my field and select my favorite plants. Adaptation to specific environments, especially in hemp, I think is going to be really important as we move forward. And those complex traits involve a large number of small effect genes. And really, that work is going to be much better done through traditional plant breeding. Traditional plant breeding also kind of built into it is the ability to select for uniformity, which is another important trait that's often lacking in hemp varieties. So the challenges or the limitations of traditional plant breeding, you're limited to the available genetic variation, right? So you can only select what you have in front of you. Whereas with gene editing, you may be able to create something novel. So if you're interested in, let's say, botrytis resistance, but your population isn't segregating for botrytis resistance, you're not going to make progress on it through traditional plant breeding. So it's also slow. Product development cycles can take 5 to 10 years, sometimes longer depending on the crop, the biology of the crop, and how many generations you're able to turn. Whereas gene editing is really quick, right? I gave an example of us taking something from a concept to seeing it in the field in 18 months. I'm not able to do that through traditional breeding. It's very precise. Gene editing, we go after one specific change. We know what we're looking for before we find it. And very often the outcome is very predictable. Genetic variation is created rather than found, right? So the example I gave of botrytis resistance, through traditional breeding, you're only going to make progress on traits for which you already have variation in your population that you can select on. Whereas with gene editing, you have the ability to create that variation. So it can be a powerful tool. And it allows you to quickly leverage new genomic information, even from other crops, right? You may see a paper published with information on a trait that's controlled by a gene that was identified in Arabidopsis, right, which is a model species. And maybe a light bulb goes on and you realize that, maybe I could solve a similar issue in hemp by targeting that same gene family. So it allows you to very quickly apply something you've learned in another crop in your crop, making it pretty powerful as all this new public genomic data becomes available. Areas where gene editing is more difficult to apply, complex or quantitative traits can be a challenge. And this is the number one take home. Your product is only as good as the germplasm you started with. So you're not going to take a variety that's not performing in the field and add a trait through gene editing and expect that variety to magically become what the grower needs. So really, I see gene editing as a really powerful tool that is going to be layered on top of and synergistic with traditional plant breeding. So hopefully I made the case to save my job. So potential targets for breeding and or gene editing. So things I'm interested in hemp: value-added cannabinoids, reduced THC production, potentially novel terpene profiles, improved flower-to-leaf ratio, disease resistance traits, modulation of flowering time for adaptation to different environments and for yield, and improved grain yield and quality. And I would love to hear feedback from the audience on traits or areas or pinch points for them, things that new varieties could bring that would add value for the grower. I would love to hear that. That's the spiel. Thank you, Robert.
Jeff, thank you.
Questions coming in?
Well, thank you again. That was a wonderful presentation. This is a very complex topic, but hopefully you've educated us all on the complexity of why the genetics are, so varied here in the hemp market. So your last slide speaks about great traits we'd all like to see. When do you expect us to see those kinds of things in commercially ready hemp seeds?
Well, and I'm not going to speak for Green Venus specifically, but for the industry as a whole, I think these things will come gradually and not all at the same time and not all in the same variety, unfortunately, right? As the baseline genetics improve coming from a lot of companies, you'll see examples of individual examples of these traits becoming more common, and over time, combinations of these traits. So value-added cannabinoids, we're seeing that. This is a constantly evolving space, and I expect gene editing to accelerate that.
So that's great. Another question. One of the challenges with our industry is we're a bit backwards. One approach would be look at an ailment out there and then try to determine what cannabinoids have an impact on that ailment and then go breed plants for the ailment. Right now, it seems like we're just breeding without really knowing exactly the cause and effect behind ailments out there. Speak to that a little bit, and how can we maybe right things?
That's always a challenge, right? Because plant breeders are not the people that are doing medical research, right? So we have two very different groups that really have very little overlap in their daily lives, kind of addressing two pieces of this puzzle, right? So I think this is something that largely will work itself out naturally over time as laws relax and allow for more medical studies. There will be more, and we're already seeing more and more research on cannabinoids in the medical area. I would expect, but I would not necessarily expect genetics companies to be behind that research. It largely will remain very separate, I would imagine.
Doesn't it seem like those two groups have to talk to each other and work, or are they in different silos and that'll never happen?
Well, no, they're not entirely siloed, but to some degree, it's a chicken and an egg situation, right? If you don't have varieties that are accumulating large amounts of some of these cannabinoids, you can't run the study. Like, where would the material come from, right? So in a sense, I would encourage plant breeders to explore all of the novel variation they can find and try to fix it. Because without that, that's the first step. That has to precede anything else that's going to happen.
Great answer. So here's a good question. You've listed 1, 2, 3, 4, 5, 6, 7 great targets that you're trying to achieve here. How will breeders prioritize which traits to work on and while maintaining low THC levels?
That's a good question. I suspect that's partially a kind of strategic commercial decision, probably somewhat regional, right? Depending on where the breeder is breeding and their environment, and probably some personal choice as well. I'm in California. We have Mediterranean climate. We have a long fall with lots of time to mature outdoor plants with very low humidity, low disease pressure, right? So you could see somebody in Washington State making very different selection decisions from me, even if we maybe started with the same segregating population. So, I would expect that alone would drive some of this regional adaptation that we're talking about. And it would be very natural for different companies to have strengths and weaknesses with regional adaptation, where their germplasm may be an excellent fit in one area and not as good of a fit in another area.
Well, it seems to be a consensus. You're getting a lot of bravos and positive accolades on the excellent presentation you've made. So thank you very much, Jeff. That was very informative. It was very content rich. And again, you were able to convey a lot of information in terms that are pretty easy to understand for the non-geneticist. So we really appreciate you being on the webinar series today.
Thank you. It was a lot of fun. I appreciate the opportunity to do it.
This was recorded. We will post it next week. Marguerite Bolt from Purdue Extension Services will handle that. And you can always go to our website at hemplitfarms.com. There'll be a link there. We appreciate you all attending. This brings our today's webinar to a close.